Navigating GMP Annex 1 updates can be complex, especially for small batch, high value products like ATMPs, orphan drugs, and personalized medicines. This webinar offers practical guidance for applying Annex 1 in real world fill finish operations, focusing on aseptic, filling a small volume liquid, and powder products. You will learn about contamination control strategies, environmental and process monitoring, equipment design, and reject handling to ensure compliance without slowing production. We are joined by Tracy Moore, Principal Consultant at TM Pharma and former MHRA Inspector, who will share insights on regulatory expectations, risk-based compliance, and quality management approaches. This session will also include a live demo of 3P Innovation's RoboFill, a small batch aseptic fill finish system, facilitated by David Johnson, Sales and Marketing Director at 3P Innovation, highlighting how automation and isolator-based design can help manufacturers meet Annex 1 requirements with confidence and control. I'm your host, James Crosspayne with Pharmaceutical Online. I'd like to thank you, the audience, for attending today's event. This is live, so we do encourage your participation. Take a moment to get comfortable with the tools on your screen. Our presenters have provided resources for additional learning below the slides. Their contact information is to the right, and below that, a question box. Please share your thoughts with us during the presentation. Tracy and David will answer as many questions as they can, and any we miss, I'll pass along for follow-up after the event. This question box can connect you with me if you run into any trouble, but please try refreshing your browser first, as it will fix most common issues. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. Now, without further ado, let's begin today's presentation. Welcome, David.
Hi, James. Yeah, thank you for the lovely introduction. And just to echo James's thoughts there, thank you for joining Tracy and I today. And to start with, obviously, James has run over some of the topics that we're going to discuss. So without further ado, I will move forwards. So for those of you that have never heard of 3P Innovation, I've got a couple of slides just to give you a quick introduction to us and our business. But we are a UK-based manufacturer. We've been established for nearly 20 years, so we'll have a big celebration next year, hopefully. But we've been operating in the pharmaceutical and life science space now for over 19 years, supporting novel drug delivery systems and aseptic fill finish for liquids and powders. And one of the reasons why we're keen to talk on this topic, as you're probably aware of, is the Annex 1 is now live and being a living, breathing regulation. And we were one of the first manufacturers to actually design from the ground up a fill finish system post-release of Annex 1. We feel this is important because we're not trying to retrofit or re-engineer existing systems that were previously designed. We've taken the Annex as it is and said, right, with a blank sheet of paper, what does a filling system need to do in order to meet with this? And today we've got an example to share with you, which will form part of this webinar, which is a powder filling process. So slightly unique. So hopefully that brings a bit, another angle of interest for you of how you do aseptic powder filling, because it is generally a niche topic. In terms of 3P Innovation, as I mentioned, we are UK based, but we do have a global reach. We have partners across the world, and we have installed aseptic fill finish lines all the way from Japan right across the globe into North America. So you may not have heard us. I often think that we're probably the industry's best kept secret, but we've been doing this with some of the most challenging products to fill and finish for nearly 19 years. And hopefully we're a brand that you'll see more and more. In terms of our portfolio, we typically start small. We start with our Discover range, supporting drug discovery, and then we try and scale our equipment with you through the clinical phase right through to commercial production. So, and what we've done is try to label some of our portfolio equipment to match those activities. So clearly there's Discover range is supporting the drug discovery part. Explore, that's really looking into how we explore the therapeutic effects of the drug during the clinical phase, and then Evolve, that's moving into production. And what you'll find is we're not like some of the very big organizations that are doing bulk filling processes. We typically specialize in the small volume, high value sector, where you're typically using ready to use container formats, and you're generally handling high value products for small patient populations. So just to start off, I guess we need to touch on some of the trends in aseptic fill finish. As I mentioned, as an organization, we work a lot in the small batch, high value drug element. This is being heavily driven by a move to sort of meeting unmet patient needs. There's been a lot of drivers from the regulators to encourage big pharma to look into orphan applications where there's currently no solution to help the patients. So you're generally seeing a faster route to approval for some of these products, which can be attractive. The swing side of that is obviously you have a much smaller patient population to cover the cost of the development. So this ends up with this situation where you have some very high value drugs for smaller patient populations. The other trend that we're seeing at the moment is a move to help drive down the cost of medicine through patient administration. So rather than for some of these novel gene therapies or cell therapies, often patients need to visit a hospital where they're going to have the product delivered via an IV, via infusion. There's a drive to try and deliver these via auto-injectors. So what we're seeing is the concentration of drug increase, viscosity of the medication increase, and then it's going into pre-filled syringe or cartridges for auto-injectors. But this poses its own challenge from an engineering perspective, as we're trying to handle a highly viscous drug, which is also can be highly sensitive at the same time. The other piece that we see from a trend perspective is more macro trends. We all remember COVID several years ago, and there was a huge demand on vials. So if you are trying to procure vials or stoppers or crimp caps, you saw a massive pull on the production of those facilities, but also there was a massive demand for lines to fill vials. That demand hasn't really gone away, but what we do see is we've seen the rise of the GLP-1s, and now we're sort of seeing this shift, whereas before vials probably made up 50-60% of the container formats. We're now seeing a huge growth in pre-filled syringes as we try and meet that demand for that particular type of product. Quite staggeringly, I saw some data the other day, and the research was suggesting that GLP-1s will account for nearly 9% of all parenteral products by 2029, which is a staggering figure when you consider all the vaccines for all the applications that we produce globally. So it really is a staggering number for one type of therapy. But in terms of what we do in the major trend that we're focusing on, we're looking at ATMPs, or advanced therapy medicinal products, the rise of RNA vaccines. I know it's quite topical. It's in the news quite a bit at the moment, thanks to our friends in the States. But also things like monoclonal antibodies and ADCs as well. A lot of these pose a lot of challenges, but they're typically focused on smaller batches. Okay, so I'm going to pass over to Tracy to talk about this slide.
Thanks, David. Okay, so this slide talks about Annex 1 at a glance, a new landscape for small batch, high value production, and what has changed. So I think this very much depends on who you speak to and in which territory you are situated in. There's definitely a stronger emphasis on contamination control strategies, but the main focus for me is about the focus on design. Robust design of facilities, equipment, processes, And the updates also brought along the ICH Q9R1 philosophy, quality risk management concept. And we're going to talk a little bit further about that later. But for me, what's changed as an ex-MHRA inspector, I think it's just clarified a lot of points that certainly in the UK we were inspecting to, but that isn't potentially true across the rest of the world because Annex 1, as written in EU GMP, was not being used by them. So there's many facts in there that I think other parts of the globe are seeing very much as a change and requiring to make some significant changes to facility and equipment in the process. Okay. So that said, for the people that have legacy equipment, I see quite a lot. I saw a lot as an inspector and now as a consultant. In fact, even recently, as yesterday, I was talking to parts of the world where they still had what they're calling traditional fill lines, which is people inside curtained areas with a fill line, which they have free, unfettered access to. So clearly with Annex 1, that is no longer compliant. And I have a personal dislike of curtains, mainly because they still allow for unfettered access. When we talk about a barrier system, that means a hard, rigid barrier. So that prevents the person's body entering into that grade A critical zone space. And there's a number of problems with retrofitting of these lines, because as you can imagine, when these were designed, they didn't have the restriction of the arm's length. So when people were designing these lines, they were thinking that it was fully acceptable for people to be able to lean over, to reach, those kind of things. Whereas Annex 1 has been very clear, A, about the barrier system and the need to remove humans away from that open processing, but 2, about the interruptions associated with first air and first air principles. So how do you do that? Well, effectively, you have to have a look to see whether that line indeed is even suitable for retrofitting. Most companies that I've spoken to that are trying to do this are trying to retrofit wraps around their lines. But I think you very much need to think about the product you're filling, the territory that you're going to try and market the product in, and understand whether that is actually feasible and whether that retrofitted line would be compliant. And certainly one of my more recent clients took the approach that because the line was designed, 20, 25 years ago, and even to try and retrofit RABs around it, it just wasn't going to be compliant because of the overreach or just the interruption of first air that just couldn't be satisfactorily resolved. So they actually did have to go into basically buying a new line. And when you look, well, they did the analysis and they were basically saying that the time to do both was essentially the same. Because you think about retrofitting and even installing RABs barriers onto old lines, you still have to completely repeat all of the qualification work, including the aseptic process simulation for the operators. If you have a number of aseptic operators that are qualified for setup, you have to repeat that for all of them in order for them to then commence. So in some respects, I think there has to be an analysis between, do you try and retrospectively fit on an old line or do you move to a new line? And different companies will have different analysis that will drive that outcome. Apologies, let me move the slide on. So in this slide, we talk about the importance of risk assessment and the avoidance of over-engineering. So I think my first point on this slide is that where it talks about from compliance to confidence or risk-based approach, I don't want you to think about that as we are going to put together a justification to say why we don't need to do something. That's not what this means. And those of you that are familiar with quality risk management will know that risk assessments describe the risks associated with the process or the equipment, whatever that risk analysis is for. It identifies hazards, risks, and identifies any residual risk that either needs to be mitigated or could be accepted. And how you bring this in so that it's a compliant approach is, once you've completed the risk assessment associated with the processes, and you identify some residual risks, if that is something that fundamentally does not meet Annex 1 requirements, then the expectation is that you will consider a redesign. And where that isn't possible, say you have a product that potentially is in short supply and could produce a market shortage, then you need to be thinking in terms of short-term mitigation and long-term changes. So it could be that when you identify these things, we try and put together mitigation in place. And where do you record all of that as a strategy? Well, that's where the contamination control strategy comes in, that explains why you're doing what you're doing and why it's acceptable, or what that long-term plan is to mitigate those risks. And I think the key to it all is that, just keep it simple. Remember the key requirements. There are basic design principles that are called out in Annex 1. And the example I gave before was, avoiding fast air, interruptions. There are common challenges such as things like the older lines with fixed hopper bowls for stoppers and removing them to be sterilised. That is very clear in Annex 1. It's an indirect product contact part and therefore should go through a double-ended autoclave and be sterilised. Bearing in mind that it also has to be cleaned first because there will be residual silicone on that hopper and track. And when we think about avoiding over-engineering, what would be nice is that when you look at Annex 1 and you think about this with in mind, can equipment manufacturers be designing the equipment so that, you don't have such problems? I remember when I first started in sterile manufacturing, which is, over 25 years ago now, the engineers having to lean over the line with their spanners and fix things. It'd be nice for things to develop where, that extensive manual interruption into that grade A is just stopped. So for me, when it talks about risk-based approach, it just basically comes back to fundamental design principles. We risk assess a really good design. Hopefully we don't have to mitigate any risk, but either way, we record it all in a contamination control strategy. Okay, moving on to contamination control strategy. The regulators do expect that this is a document that provides both you and them with a roadmap. And you should be able to provide this to an inspector or even a new member of staff, and it clearly detail how your processes are operating. And where it says about integrating CCS into everyday operations, it should be, the CCS has to reflect what you're doing. That is the kind of what the inspectors will take away on how you are controlling the contamination, and that's effectively how you'll be inspected. So it does need to be clear. It does need to reference the risk assessments, the procedures, the current versions, so that they can pick those up easily. And in each part of that, have a very small summary for each section so that it just summarizes what you found. And if you keep it kind of simple like that, you shouldn't go too far wrong. And I think I now pass back over to David.
Thank you, Tracy. Just talking about the contamination control strategy. Obviously, it's a very wide-ranging document. It covers much more than just the fill finish line. But specifically, when we are looking at the fill finish line, one of the major things are environmental monitoring. And more sort of nichely, or specifically for today, we were going to talk a little bit about aseptic powder filling. And the key reason why we sort of flag this as a topic is one of the things that you're monitoring for are any particulates. We all know the reason why we're monitoring for particulates. But when you are trying to put powder into a vial or a syringe, it's inevitably, it's a dust making process. If you take from powder and you pour in, you will get a cloud of dust. Well, hopefully not a cloud of dust, because it would be a nice controlled technology. But if you consider, if you were making a cake at home and you took the flour in a bowl, you get a big cartoon cloud of powder. So if we are monitoring, we're real time monitoring the particles, clearly we're going to get readings on a quite frequent basis if we're doing powder dosing. So, and this is one of the areas really where you need to get involved with and engage with your regulator and talk around your monitoring process. Now, it may be that with the technology that you're employing and with your particular powder or your particular characteristics of that formulation, it may be inherently not a very dusty powder. And I'd like dustiness to be more of a scientific measure for this piece, but unfortunately it's not at the moment. But you may be able to run a normal monitoring process, just as you would with any type of liquid filling process. So this is your monitors are running during setup, during your operation, during the whole process, you are monitoring and you're building up that big body of data. In the instances where maybe the product is quite a dusty product, or the technology does create dust or airborne dust, you're going to quite quickly start to see these alarms going off. The count is going to be picking up the dust that you're producing. And this is where you need to change your approach. And while the approach seems quite simple, it is you run the monitoring right up until the point of filling. We pause monitoring, we fill your product, and then we restart that monitoring process. The key thing here is data is king, and this is probably something that works for more than just monitoring, but having that data of pre and post powder filling is really essential, so that you can try and create that understanding of, in the normal conditions, this is sort of level that we're expecting to see. When you are powder filling, you pause and then restart as soon as possible, and then it's try and get that discussion, that piece on whether that approach is suitable for your product with your regulatory partner on that filling line is really essential. And part of that will be making sure you are monitoring at your high risk areas. Again, not too dissimilar, well, not as dissimilar at all from a liquid filling. You should be by regulation measuring your highest risk areas. So that's identifying where you're most likely to see your open vials, because that's where you definitely don't want any particulate contamination. And we'll try and show you.
Sorry, David, I was just going to support you there. Just in case people weren't sure of the clause in Annex 1 that says that that's permissible. There is a section there in 9.20 that talks about where effectively, if the product is going to damage the particle counter or present a hazard, then the frequency and strategy employed should still assure the environment classification both prior to and exposure to the risk. So apologies for interrupting. I just thought it was probably important for people that aren't familiar with that paragraph, just to give it context to what you're saying.
Yeah, no, I'm pleased you did. 'Cause the other thing that's worth doing is, if you have a PDF version of the annex, search powder. And unfortunately, it doesn't come up very often. So it is an area where people are unsure, and they do have a lot of questions about it. So yeah, I'm pleased that you've jumped in there with a bit of guidance. Okay, so we will move forwards, and equipment, isolators, and automation. So a key topic there is isolator versus RABs. Earlier on, we heard Tracy mention around some, if you like, traditional filling lines where people still employed curtains, for example, as barriers. And I think here the Annex is quite clear. We need to move more to that physical barrier. But I think there's still this argument around what really is sort of best practice and what we should be looking to do. And that is always isolators over RABs. And I'm quite pleased in this instance, because actually it's not very often where maybe regulation supports a financial business case for investment, but actually here it really does. And if you look at a typical filling suite that's based with a RABS line versus a filling suite that's going to utilize an isolator type technology, the first and most obvious benefit is the room setup. So quite clearly, if you go through the room grades, you can see that an isolator system can be situated in a grade C room, provided you use it in the right manner and have the correct cascading of chambers around your critical zone, around your filling area, versus a RAB system. And actually, when you start looking at, yes, an isolated system clearly has a higher upfront investment cost than a RAB system. But actually, when you start looking at things like the actual operation of that filling room, the air management system, the energy consumption of those systems, actually, when you're investing in a filling line, you want this filling line to be running for a long time. It's not a short-term investment. You're going to want this running for 10, 15 years. And when you look at the lifetime cost of managing a grade B room versus a grade C room, it's quite staggering the amount of money you would save on energy consumption. So there's a financial benefit. On top of that, you've also got, as any good employer will be trying to do, is trying to keep their operators happy. And Tracy, I'm sure you've been in plenty of grade B and grade C rooms. I know I have. But the gowning procedure to go into grade C versus grade B is very different. It's another level. And so that's one thing: the amount of time, the amount of gowning, and ancillary items that you just consume on a daily basis. But also just how pleasant is it for your operators to be working in those conditions? So you can actually, your operators are more comfortable working for longer periods of time. A happy workforce is ultimately what we also want, as well as happy patients. So I think this is a real positive, where regulation is promoting a higher level, a higher quality system, which is going to improve the product quality to the end result to the patient. But also it provides the opportunity for businesses to invest with confidence that actually there's a return on the investment here. We're going to see savings versus what we have done previously. So not only is it a better way to produce medicine safely, it's also a better way to produce medicines cost effectively and improve the conditions for your own workforce. So I think it ticks a lot of boxes here. So while there's been a lot of focus so far on retrofitting, I think moving forwards, if it's a new greenfield facility or you're looking to upgrade and purchase a new line, moving towards an isolator has a lot of benefits versus a RAB system.
And David, if I just come in on that,
Yeah.
just with regards from a regulator's standpoint, providing isolators are operated correctly, inspectors absolutely inherently know that this is a safer environment for products to be manufactured. So if you have the choice between a RABS and an isolator, I would definitely consider future-proofing yourselves and moving and, take the plunge and purchasing the isolator. And I was at some conferences last year where US FDA policymakers quite clearly said that certainly for them, they wouldn't be accepting any new applications for any product that was being manufactured in an open RABS. So yeah, there was absolutely a drive from the regulators to push it forward.
Yeah, I think there's a couple of thoughts there as well. There's regulation only goes in one direction typically. So while we're here talking about the latest Annex 1 release, it's exactly that. It's the latest release. There will be a future release, and it will have the key objective will be to drive further the quality control around producing safe medicines. So expect the next one to be even stricter. So again, you want to make a sensible future investment choice is to go for isolated based systems. So it just makes a lot of sense from my perspective. And I think often you hear, and this is not just maybe linked with isolators, but just generally when it comes to quality driven decisions, if the technology exists, then why aren't you doing it, right? Because you have a choice to produce medicines at a particular quality and safety level. That should be your priority. You should be trying and make sure that you're operating at the highest level, because this is what's going into people's bodies at the end of the day. So not choosing something, if it's there and available, is not typically a very well received message to regulators.
It's actually, you can take that a little bit further, David. There is actually an article in the EU Directive 2001/83 that actually says that marketing authorization holders must keep up with scientific and technological advancements. And if we think that isolators have been around for 20 years or more, then, clearly it's written in the directive that it must be done. But as we're seeing, there are people further behind in this journey.
Yeah, I agree. Okay, so I'm now going to nip to our facility floor, where we've got a camera set up to walk you through one of our units that are being prepared for a customer, and we can see some of the pieces that we've talked about today. So I'm going to dip out now and leave you with Tracy and James while I run downstairs.
Thank you so much, David. While he's getting set up for the live demo, allow me to draw your attention to a few questions our presenters have asked of you that are just below me. Please take a moment to share your organization's readiness for Annex 1, what area you are experiencing the biggest challenge from as a result of Annex 1, what containers your site will be using in the next 5 years, and what your choice is for the most important consideration for filling line purchases. We appreciate you taking the time to answer. Now allow me to hand things off to David for the demo.
So welcome to the facility floor at 3P Innovation, and behind me we have our RoboFill solution, which we'll run through some of the points that we've discussed. But first, just quickly touching on the last topic of RABs v isolators, and to try and make it more real, if we were using a RABs-based system, this would be based in a grade B room. And as you're probably aware, to operate in a grade B room, I would have to have full gowning, hoods and mask on. If you ask some of your operators, it's not a very pleasant way to work, particularly if you're doing full shift. With an isolator-based system, we can actually position it in a grade C room, so it's much less gowning restrictions on the operator. It's much more pleasant to work in. There's also less gowning required, so all that time changing in and out of gowns, the waste of the gowns is much more reduced. And actually, with an isolator, if you operate as a closed isolator, which might be possible for very small batches, you could actually run it in a grade D environment. So we'll go and have a look at the machine. I'll give you a quick run through on how it basically works, and then we will pick up some of the key specifics around Annex 1, your CCS, and sort of your environmental monitoring, reject monitoring, etc., etc. Okay, so come with me. So a quick introduction to the system. It's the automation, the filling system, and the isolator are all done by 3P Innovation. By having everything done by the same design team, we're able to make a very compact unit with everything housed within this system. There's no external HVAC system up in the roof. There's no remote control panel or electrical cabinet. Everything is based in this system. And obviously to operate and service it, it's all done by 3P Innovation. The system's designed to handle anything that's ready to use. So in a ready-to-use tub, you've got your vials, syringes, cartridges. Even if you have a novel delivery device, provided you can get it in a tub, we could handle it in this system. It's also designed to do liquids and powders. So this system here is for powders. However, I'll show you how it could be configured for liquid filling. And if any of you have combination products or dual chamber devices, this could do liquid and powder filling on the same line. So how does it work? Well, it starts with the ready-to-use container format, the tub, which typically comes double bagged. The operator is going to remove the first bag and load the tub in through this mouse hole. And you might make out in the image here that you can see there's a couple of tubs already there, ready to go in their inner bag. They move down the conveyor, the operator preps the bag, and then the machine grips the bag, cuts open the bag and pushes the tub through to the next chamber, which, if you follow me, is what we would call the delidding chamber. And surprise, surprise, what we're doing here is we're removing the lid from the ready-to-use tub and the internal liner. The first thing that happens is we apply a heat to the top of the tub. And what we're doing here is we're warming the adhesive. And this is so when we peel back the lid, we don't release any particulates into the environment. So the robot will peel the lid back at a specific angle, and then it will then remove the liner. And all this happens without anything moving over the top of the open vials. Once this is done, the tub moves into the filling chamber. So as I mentioned, this filling chamber is for filling aseptic, aseptic powder filling. So there's quite a lot going on, but the general operation is the tub comes in. The first thing we do is we remove the nest out of the tub. So here you can see the nest of the vials. And then during the filling process, we take each individual vial to the filling station where we fill the vial, stopper the vial, and then it gets returned back into the nest. This individualization of the vial is a very key important of allowing us or enabling us to have multiple quality inspection points throughout the filling process. So first thing we're doing is we're making sure there's a vial present. Then we're filling to a known weight. So we're measuring the weight of every vial dosed. And then we're placing a stopper onto the vial. And then we're also having visual inspection on the stopper height and the stopper's level. So all that happens before the vial goes back into the nest. The tub or the nest goes back into the tub, and then it will move along into the crimping process. So let's move to the crimping chamber. It's a bit easier to see what's going on in this one. So as the filling chamber, tub comes in, nest comes out the tub, and then we individualize the vial by removing them one at a time and placing them on our crimping station. We use a rotary crimp, and this is where we spin the vial with a passive crimp wheel, and this ensures there's no relative motion between the vial and the crimp cap. Again, it's shown to be reduce the level of particulates generated with this process versus jaw crimping, for example. It also lends itself to a few in-process quality checks, namely monitoring the compression force. So we measure the compression force applied to every single vial. But also, as the vial is spinning, we can do a visual inspection on the crimp quality. So we can inspect for any known sort of imperfections, and then we can isolate that vial by placing it in a reject rack in the bottom corner. Once all the vials are crimped, we go back into the nest, back into the tub, and it moves down the line out into the end chamber, which is an exit RABS. So I'm not going to show you that because it's not as exciting. What I do want to do is go into some of the details in the crimping chamber and the filling chamber that are more relevant to some of the topics that we've talked about so far today. In terms of having a robust CCS that covers more than just your filling operation, obviously it looks at your supply chain, your operation, your gowning procedure. So having a simplified gowning procedure can really help. But it also looks at how you set up the system. And that's one thing that we've taken into account. Now, for a line such as this size, which is going to do anywhere between sort of 1,000 to 3,000 vials an hour, it's really geared towards that high value, low volume throughput of products such as ATMPs, monoclonal antibodies, gene therapies, for example. So in terms of making sure we feed the right number of stoppers and crimp caps into the system to meet that throughput, we don't necessarily need a vibrating bowl. In fact, if we can remove the vibrating bowl, then that's a benefit. Reason why it's a benefit? Well, vibrating bowls, they can create particulates because there's a lot of metal on metal, but more from a contamination point, a setup, and from an operator perspective. They're product contact. They have to be sterilised, and they're sterilised offline and brought to the line. So they're going to come in a bag, you're going to open the door, load in the system, close the door, run a decon, and then through gloves you're going to have to debag the system, load your bowl in. It can be quite a challenge. So we've done away with that altogether. And what we use actually is a vision-guided robot to pick up stoppers and put them on the vial ready for crimping or stopping. And the way that we do this is we have a small vibrating tray. So I don't know if you can see in here, it may be that we have a little video also running of this, but we have a small tray there which vibrates. So the stoppers or the crimp caps are loaded on here, and as the tray vibrates, they separate. And this allows the vision-guided system or vision-guided robot to pick them individually and load them onto the vials. Why is this better than a bowl feeder for this size of system? Well, A, it's not size dependent. The tray can handle stoppers of any sizes, but also it can be sterilised offline and it can be brought to the line and loaded via an RTP canister. So you've got an RTP port there, so you could sterilise it in your autoclave, load it into an RTP canister, bring it to the line, and we can install it without breaking the containment of the isolator. So it's much easier to install in a much more compliant manner. So again, we're reducing the risk of any sort of contamination. We're going to go through to the filling chamber next. And like I said, much more going on in the filling chamber. One of the key things that we've talked around is first air principles and airflow. And obviously this is a powder filling system at the moment. It's got our filter weight, which is a proprietary 3P technology. It's ideal for handling challenging powders, but it's also ideal for aseptic powder filling into vials and syringes especially. But it relies on gravity. In fact, most powder technologies rely on gravity to fill, and which means we need to have something above the open vial, which is our critical zone. So typically in a liquid filling station, you wouldn't have any of this. There would literally just be the minimum amount possible above the open vial, the filling needle. Here we have to have a filter weight head, because this is what's going to dose the powder in. So how do we manage that risk? Well, we manage the risk with some CFD work before. We do a lot of smoke studies during the commissioning phase and the design phase to ensure that the air is still free to flow around the vial and, most importantly, away from the vial without breaking that laminar flow, without becoming turbulent. So that's a critical piece to demonstrate, and that's something that regulators will want to see before you can qualify your line. We've got, similar to the crimping chamber, again, we have a lot of inspection. So we have the weight inspection. We also have visual inspection on the stopper. So we're inspecting the stopper height and the stopper's level before the vials go back into the nest. Again, if they don't, they get placed in the reject tray where they are quarantined, and then they can be then managed by the company. This is a much better way of doing it than systems that typically fill directly in nest, and then they guide the operator to manually remove any vials that are out of specification. Obviously, you can clearly see how that increases the risk of out of spec vials or syringes making their way into the supply chain. Another element to consider that we've talked around is environmental monitoring, particularly particulate monitoring. So we have got our isokinetic probes at sort of key areas. First one, there's one here right above the nest of vials. So here, why here? Well, when all the vials are open, This is a critical space, and we want to ensure that the air going over it is clean and there are no contaminants. It'd be very easy to put this somewhere else and get some really nice results, but actually it needs to be at the highest risk areas. And it's also, we have another one positioned back by the powder filling station. Again, because they're situated as an open vial, and we want to make sure that the air there is clean without any contaminants. The other element to consider with isokinetic probes is where you position the actual counter. And you'll see they have tubes going down and into the base, and there we have situated the counter. And so by keeping the tube length as short as possible and actually reducing any bends in the tube, then we're going to get a much better, more accurate reflection of any particulates that are detected in here. With longer tubes or with more bends, it's more likely that any particles that are going through the filter collect on the inner walls of the tube and actually don't make it to the counter, which is going to obviously something we want to avoid. I think that's probably covered some of the key things that we wanted to or have spoken about today. What we will do now is talk around actual implementation in the real world and what that looks like. Before that, there's going to be a quick polling question, and that's going to let me have time to get back to the desk to do the presentation. So thank you for allowing me to show you our system, and I will go and join up with Tracy again.
Excellent demonstration, David. Thank you for going the extra mile to share that with us. While he makes his way back to the presentation studio, if you haven't had the chance, please take a moment to answer the questions below me and share your experiences with Annex 1, containers, and filling lines. Now let me hand things over to Tracy to share best practices for Annex 1 implementations.
All right, thank you, James. So best practices in Annex 1 implementation. So I'm sure everybody's already done this now, but obviously the first step that most sites were doing was a gap analysis. And from that gap, which from the formats that I've seen can be, multiple sheets on an Excel spreadsheet, line by line, to just general high-level topics that companies feel are a risk. Whichever option you chose, I'm sure there will be things to done even in the most compliant of sites. But what I want to draw you back to is I want to draw you back to thinking about, what Annex 1's intent was, which was to ensure, the design of your facility equipment processes are robust and compliant within the kind of essence of Annex 1. So what does that mean? It means that when you're putting a process in place into a facility, then in an ideal world, you will start with how the process is going to be performed and you would design the facility around it. Now, clearly, there are some things that are required, certainly for the HVAC, for example, to operate correctly. What process is going in there? Is it going to be a small molecule? Is it biological? If it's a biological, what BSL level do you need? Is it just cascade that's needed or containment? Is it a positive isolator? Is it a negative isolator? How many pairs or airlocks are going to be needed? How are those material transfers going to occur? And in order to know how the material transfers are going to occur, then you need to know what equipment you're going to be using. Things like, David talked about vials earlier. The stoppers, are they going to be sterilized in vessels and then automatically lifted and fed to the line? Or are these going to be provided by the stopper manufacturer in bags that are either ready to use, that have RTP ports and docks that can easily transferred into the loading hoppers? Or, are they going to be ready to sterilize? So they're going through either a gamma or an autoclave cycle. So there's lots of things needed in order to get the design of that facility correct. And I think certainly if you're starting from scratch, it's ideal, but know the process first. we talked about retrofitting of lines, retrofitting of a process into an already designed facility is equally as tricky and as difficult. But where we talk about the design and having to apply Annex 1 retrospectively from a facility perspective, there are certain checks and balances that you can do. So when that gap analysis was performed, hopefully it did go back to the design, and where it wasn't, probably my advice to you is just go back and check some key things with regards to certainly design of the facility, design of the equipment, with regards to Annex 1, design of the processes, even down to how the material transfers are occurring, and the number of personal airlocks that lead to that clean room that's going to be performing the processing. One of the clarifications in Annex 1 was that those personal airlocks should be a grade stepwise increase before finally reaching the clean room of the processing, and that should obviously match the same grade upon entrance. So there's a number of things there to think about, and I think probably the biggest key for me. In fact, I recently did a talk where I analysed the number of hits in Annex 1 that design had, controls had, and monitoring had. And I've been present at presentations where people have said, well, monitoring has the most hits, therefore monitoring should be the emphasis. And that's what's meant by Annex 1. I've also heard people say, well, no, QRM is, the first part of Annex 1, so QRM should be the focus. We should be risk assessing and making sure that we can risk assess and not necessarily do what Annex 1 is saying. And these things are all misleading. That's not what the intent is at all. If we think about proper GMP being, we design things to comply. We then control those design elements. And then the monitoring is what gives you your assurance that the design is correct and your controls are right. I think the phrase I like to use is you can't monitor or control your way out of a poor design. The robust CCS that you need to be presenting is based on those fundamental principles of design, control, and monitoring, but with the design being the essence of it. And by control, if we think about Chapter 5, Section 5.21, the technical and organisational measures, they weren't repeated in Annex 1, but you need to be going back to the different chapters to see what those controls and measures are and implementing those. And then the monitoring, obviously, just provides that assurance. So that's your personnel monitoring, in-process monitoring, materials, income and testing, those kind of things, environmental monitoring, utilities, process monitoring, and then obviously finally aseptic process simulation, where you have an aseptic process. So it's that's how you implement Annex 1 in line with best practice, in a nutshell. Okay. Hopefully that wasn't too garbled. And David, I'm hoping you're back from the facility floor and you're going to do a recap for us.
I think in terms of a recap for what we talked about today, I can appreciate some of this. You may have gone over it before, but essentially that's because it's important. And hopefully there are other bits that you can take away and implement into your own processes when it comes to looking at Annex 1 compliance. From my side, I've hopefully sort of highlighted some of the differences between liquid and powder aseptic filling. I hope you enjoyed the look around on the RoboFill system. If anyone does want to go and have a more closer look, that unit will be with us for next couple of months before it ships. So you can reach out independently and come and have a look at that if you want. And I think the other piece, I think we've got some Q&A coming up. But if there are any other questions that have popped up that we don't get a chance to answer today, please feel free to send them to us, and we can put a response together and get back to you. And I think there's also in the resources, I think there's a link to some of the frequently asked questions that we've already tried to answer. So I think we'll move to the final slide. James, have you collected some of the questions from the audience?
Absolutely. First, let me thank you guys for the amazing presentation and just give a few reminders to the audience as well. Before the Q&A begins, I just want to remind that you can connect directly with our presenters. Their information is located on the side of your screen. And I want to remind as well to please take a moment to answer the questions they've asked of you below me if you haven't had a chance already. We also encourage you to continue submitting your questions via the Q&A box. So our first question comes from Sabrina, who is asking, with all the advancements in containment and production equipment, do you believe we're in a position to advance monitoring and testing of the product and environment? And if so, what does that look like?
Oh, wow. Okay.
Go ahead, Tracy. Yep.
Yeah, well, it's just the thoughts that I've seen certainly advancements in the kind of monitoring equipment. There's some really nice new equipment out there. I know the rapid micro monitoring equipment is certainly coming along. At the moment, I don't think anyone's quite cracked it entirely, but I don't think it's going to be too long before there is a robust solution to the age-old problem of swapping out settle plates within a grade A environment. But, this equipment's not just used for, monitoring grade A. There's equipment out there that can help facilities with things like, they've had a problem with recurrent clean room monitors. And there's some, the rapid micro analyzers can detect, can be screened on a wall and detect points of weakness within the vinyl, for example. So I'm quite excited for what the future holds in this space. And I think a lot of it will be, again, about just keeping people out of that process and it being automated. And I'm excited for what that does look like. But at the moment, I don't know what it looks like. And companies are getting there. I'm just not sure they're there yet.
Yeah. I think if I can sort of drop a couple of comments as well. I think around the advancements in containment and production, as Tracy said, you need to consider advancements in containment and production equipment alongside advancements in monitoring and measuring equipment at the same time. It's not one or the other. And just because one side's got better doesn't mean you don't have to do the other side. They're solutions that are designed or that need to be designed to work together. I think from an automation perspective and a containment perspective, there's a lot of discussion around gloveless systems at the moment. Automating as much as the process as possible. We know that gloves can, I think people maybe wrongly could just consider pinholes in gloves as an issue. But actually it's not just that. Gloves can also be quite difficult to clean, and they can get in the way and disturb airflow as well. So trying to automate as much as the process where automation makes sense is a very sensible thing to do. But it certainly doesn't remove the need to be monitoring as well. And I think the other piece around gloveless automation is, there's even in a gloveless system, you need to set the lines up. So how are you going to set lines up for filling and finish? There's still these potential breaks in containment where you could potentially contaminate the line accidentally. And then finally, there's also the consideration of if something goes wrong, being able to actually pause manufacturing and resolve any issues. So how do you do that without gloves? Or is there a feature where you have an emergency glove available? So again, it's not a question of just get rid of gloves. We've got this brilliant biofluorescence monitoring real time. We don't need to do this anymore. It's around using the right combinations of technologies. And that includes lots of monitoring, because we know when it comes to things like quality data is king as well.
Thank you both so much for that. I think we have time for one more question. So I'm going to ask this one from Marcus, who would like you to give examples of Annex 1 topics that would be different for small batch gene therapy, viral vectors versus large batch biologics like MAB.
Okay. Well, David, shall you go first from a machine perspective, or would you like me to go first?
I think when it comes to sort of machines and the sheer size of machines, clearly the biggest challenge on a larger volume line is getting things in and out of the line, because you're typically running these lines at much higher volume, and versus a gene therapy where you may be doing 5,000 batch or 10,000 batch of vials. You're typically going to be looking at something around ready to use vials, which are coming in pre-sterilized, versus if you're looking at a bulk filled line, you're probably going to be doing the sterilization, washing, and drying process yourself. So that there's a huge element there that you're going to have to work on in terms of your CCS, in terms of how you manage that stock going in, how you're ensuring that the container formats, stoppers, everything in that line is handled correctly and introduced into the line correctly, and that you can document that and measure that. So I think from a machine perspective, that's probably the main area I'd look at. So it's not necessarily the machine itself, but your ancillary items, your container formats, your stoppers, your caps, etc., and how you manage to document that. Any thoughts, Tracy?
And yeah, well, when I was just listening to the question, so it says the examples of Annex 1 topics that would be different. So I think the first disclaimer is depending on which territory you're sitting in. So for ATMPs, Annex 1, if you're in a PICS community, would be applicable because that's what the PICS GMPs say. If you're sitting in Europe, then currently it's not, but Annex 1 will be adopted into it. So that's the first probably health check. I'm quite simplistic over this. So yeah, gene therapy, small batch sizes. So the first thing is probably the APS demand, so the aseptic process simulation demand. So with small batch sizes, with manual processes, there's quite a distinct difference. So things like manual compounding, the APS is on the person on how they can do it. So in order to start that, they would have to have 3 aseptic process simulations for that actual person. And I'm not sure whether James or David said about the small volumes being 5,000. So quite often these can be below the 5,000 unit. So we are talking really, really small. And then when we think about the large batch processing, like for a monoclonal antibody, then they're usually larger volumes and on an automated fill line. So maybe not the small batch processing, and it depends on the volumes really, how companies are doing this. And the other consideration I was thinking is obviously I don't want to mention PubSit, but filtration expectations. PubSit is probably one for another day. But obviously we'll leave that on another day, I think. I don't want to cause a riot in the middle of the webinar. And then other things like, depending on, I think, did they mention viral vectors? So, what is the BSL requirement for that viral vector? That could be different. So the way the facility is designed, is it traditional cascade versus containment? Is it BSL 1, 2, 3? So there's those kind of considerations. And then environmental and process monitoring strategy. How do the companies go about getting that in place? Monitoring of a biological safety cabinet is completely different to monitoring of a fill finish line. However, the decisions that you take are similar in that you need those monitoring points to be in the locations that is going to give the right data, and the right data that someone who's going to be releasing that batch can make a decision on. So this is obviously a little trickier within a biological safety cabinet because it's usually quite cramped. There's normally, like I say, manual operations occurring in there. And inspectors will certainly home in on certainly that monitoring location point for those. And then I was just thinking about what other things would be implicated. So things like closed systems. So Annex 1, for example, the previous version didn't really touch on closed systems too much or single-use systems, but these are very much employed in ATMP manufacture and even large-scale biologics. Single-use systems seem to be the way forward. They both has positives. Well, for me, they are mainly positives. I mean, there's some downsides that people are still getting their heads around, such as integrity testing of single-use systems. But, you still have to assure the sterility. So it's still required. And I think, the starting materials is one thing. If we look at the introduction to Annex 1, it talks about anything that's used as part of a process. You should be looking at it with regards to risk from particulate, pyroburden, or bioburden. There could be other things to think about, like TSE or anything from an animal origin. There's other things to consider. So I don't think we can cover it in this webinar, if I'm honest, because there's just too much, too much to go into. But hopefully that just gives people a flavor.
Absolutely. I think that's a good note to end on, because we're going to be passing all these amazing questions the audience has asked to you guys offline to follow up. So I know there's a lot more to talk about, and I'm sure you guys look forward to that. If you want to connect with our speakers directly, please take a moment to scan this QR code that I have for you on screen. I'd love to hear from you. I'd like to thank David and Tracy for providing their expertise today, and you, the audience. We appreciate you being with us here today and hope to see you again soon. Have a wonderful rest of your day.